So welcome everyone to this seminar arranged by the Center for Future Chemical Risk Assessment and Management. My name is Slunge and I'm leading this center now since last year. The topic of today is hazardous substances in marine management. We are very much looking forward to this. We have two prominent speakers. We have Joachim Sturve here from this faculty, who is a professor in ecotoxicology, right? And Johan Gustafsson from our marine agency, who work with as an analyst on, and he can say something about how to use this type of assessment in policy making. And Johan has a PhD in environmental chemistry from Stockholm University. We also welcome our online participants, and we'll try to make the best here to communicate both with the audience here and with you online, and we'll watch the chat carefully if you have any questions. So we'll have around 15 minutes with Johan first. It can be a few minutes for questions, and then we have Joachim, and then we'll have a broader discussion.
So thank you for the nice introduction. So my name is Johan Gustafsson and I work at the Swedish Agency for Marine and Water Management. And I was invited by Joachim today to co-present this. So the idea here is that I will sort of give the framework and the context for what Joachim will later talk about. So my presentation will be much broader aspects. This is me 2.5 years ago at Stockholm University, where I defended this thesis. And at the end of my PhD studies, I sort of started thinking, now I spent a lot of time and energy on this research, as most of us probably do. And I started thinking, how will this actually lead to a better environment? And the typical answer I got in some way or another was that your studies will be used by regulatory agencies to implement measures. Sounds good. And the follow-up question from me was, but how? And then the answers were much more vague. There were answers, but not really compelling. So what I found out, and that feeling has sort of been reinforced now, is that these are two different spheres. We have academia and we have management. And there is an overlap, but there are different languages and different cultures. And this seminar, and also what Joachim will talk about later, is in this overlap. So what I already knew and know even more now is that chemical legislation is complicated. So on the left-hand side, I've listed a few of the sort of central pieces of chemical legislation. And this is mainly EU legislation, but there are over 40 different regulations in EU covering chemicals, and then we haven't even started talking about the global level or our national level. And I like this illustration of chemical legislation as a patchwork, where different patches cover different areas. There could be overlap, and in the worst case there are some holes. And I will talk about one of these patches today, and that would be the Marine Strategy Framework Directive. And just to satisfy my curiosity, I would like to make a very quick poll here. Anybody who has ever heard about the Marine Strategy Framework before, could you raise your hands? Have. Okay, that's good. About half. I would say a little bit more. Then you know much more than me than I did 2.5 years ago. So the Marine Strategy Framework, it's an EU directive, so it applies to all EU member states which have a coast. It was implemented in 2010, and the geographical area that it covers is from the coastline to the outer border of the exclusive economic zone of each country. And the aim here is a clean, healthy, and productive oceans. And what it says in the directive is that member states shall take the necessary measures to achieve or maintain good environmental status. So this good environmental status is a key concept in this directive. And this should have been done by the year 2020 at the latest. I'm not sure if you heard about the state of the marine environment, but my optimistic interpretation of this is that this is still work in progress. in the directive, it's divided into different thematic areas. So this is what's called descriptors in the directive. And on the left-hand side here, with some strange font, we have sort of the state these descriptors. We have marine biodiversity, including birds, marine mammals, and fish. We have pelagic and benthic habitats and food webs. And on the right-hand side, there are sort of the pressures which affect these states. And these are non-indigenous species, commercial fish and shellfish, so fishing basically, eutrophication, seabed integrity, hydrographical conditions, hazardous substances in seafood, marine litter, and energy, which is currently mainly focused on underwater noise. And I will talk about hazardous substances during this part. All the other ones will not be included. So under each thematic area, there are subcategories, and these are called criteria. The first criteria, so descriptor 8, criteria 1, D8C1, is level of hazardous substances. So this is typically what we think about when we think about hazardous substances. You have a level of something and compare it to a threshold. And then there's criteria number 2, effects of hazardous substances on species and habitats. So this is the criteria where Joachim's work fits in. And then we also have criteria now, the third one, acute pollution events, which is currently mainly focused on oil spills. And then we also have a fourth criteria, effects of acute pollution events. This is currently not so developed yet, but it hopefully will be. This will be interesting. So we have a 6-year management cycle, similar to the Water Framework Directive. The first step is that we should define what good environmental status is. This is what we should reach, but we need to define it in a concrete and quantitative manner so that we can say if we reach it or not. The next step is that we should perform a state assessment. We should assess the state of our oceans. The third step is to update and assess targets, and I will get back to what targets are. Then we need to revise our environmental monitoring programs, because we need to have monitoring in place so that we can assess the state. This will lead to a program of measures. So all of this that we're doing, the end point is that we should implement measures. And then, so this is producing the program, and then the last and perhaps most difficult step is to actually perform these measures. And then we start all over again. And in 2024, we finished these 2 steps. And what's ongoing work is mainly assessing these targets. And we have also started preparing for the revision of the program of measures. I thought I would just briefly try to explain how the Marine Strategy Framework Directive is implemented. It's quite complicated, I would say, when I was new at least. In the top here we have, of course, the actual directive itself. That's the core. Then at an EU level we have something called the Common Implementation Strategy. So this is different working groups which are discussing and producing material for implementing this. One of these materials could be, for example, guidance documents. This can be about anything. It can be a reporting format. It can be how you should assess the state of the environment. The European Commission can also make decisions. So this is one important decision. The Commission also assesses how the member states implement this directive. Are they following what they should or not? And then there are also tight connections to these 2 other EU directives. But in formulating this work, it was sort of recognized that a country cannot alone fix the oceans. We need to work together. So a lot of the work is done in these regional sea conventions, HELCOM and OSPAR. So these are organizations where countries in the Baltic Sea area for HELCOM, and OSPAR, it's the Northeast Atlantic, so it's spanning from Portugal in the south to Norway in the north. So these countries are members and more or less work on a voluntary basis to try to take measures to improve the state of these oceans. one common or one important part of this work is to agree on common threshold values. If one country thinks that a level of 10 of something is okay, while another country thinks that it should be 0.1, you can't really compare these assessments. So this is a platform where we try to agree on which threshold values we should use. Another very important part is to coordinate environmental monitoring. And then we have the Swedish level where we have implemented this. So this is an EU directive. It means that we have to implement it in our national law. So this has been implemented mainly by this miljöbalken. We also have something called the havsmiljöförordningen. And then FRAM, the agency I work with, also issued these föreskrifter. So very quickly, I will not go into any details of this. Then you have to invite me for a second talk. So the state of our oceans. First one is level of hazardous substances. Good environmental status is generally not reached, as you can see with all the red markings. However, this is based on 15 substances or substance groups. So the issue we have is what about the potential effect of all the other hundreds of thousands of chemicals we use in our society? So one tool we have at our disposal is biological effects. And here, in very messy order, you have the indicators that we consider. So we have white-tailed sea eagle productivity, which is mainly linked to the usage of DDT. We have Imposex, which is mainly linked to the use of tributyl-10 as an anti-fouling paint. We have reproductive disorders of amphipods, which is a bit more of a general marker. And then we also have disturbances in the reproduction of eel pout. And this is in need of development, so that's where Joachim's work is focused. So that was sort of the state of our environment. The next step in the management cycle is targets. And in the Marine Strategy Framework Directive, it says that member states should establish targets to guide progress towards achieving good environmental status. So the idea here is that it should be stepping stones towards the ultimate aim. So if you have a concentration of 100 and it decreases with 1 per year and the aim is 1, it will take you 100 years to reach your goal. That can be a bit sad. So typically, as long as we're reaching our targets, our stepping stones, we're sort of happy with our management. And if these targets that we set, we decide them. If these are not reached or risk not being reached, measures should be implemented. And this leads to the next step, that we should have a program of measures. And in Sweden, we have implemented these targets as miljökvalitetsnormer, which means that they are legally binding to follow in, for example, planning and permitting processes. So the final step in this management cycle is a program of measures. This is the ultimate aim of our work. And this program of measures should contain all measures required to reach good environmental status. Quite a daunting task, but we do our best. And this is a program of measure in Sweden according to Miljöbalken, 5th chapter. And this comes with some limitations. And one of them is that measures can only be targeted towards other agencies and municipalities. So within this tool, we can't point to a particular industry and say you have to do something. We would point to the agency responsible for overseeing that in industry, saying this sector has to reduce its CO2 emission for us to achieve our targets. And this program of measures includes all ongoing measures in other frameworks. So everything that we're doing anyway, we have to do that. But then we also add new measures, which are specifically required for the marine environment. So one example of this is OPH, Åtgärdsprogrammet för havsmiljön, number 40, which has to do about anti-fouling paints on leisure crafts. So before I hand over to Joachim, a few conclusions. This was aimed at being just a quick glimpse into how our marine management works. I can talk a lot more about all the different steps. But one message is that the comprehensive assessment of the total risk hazardous substances posed to the marine environment is the basis for implementing our measures. Without a good assessment, the final goal of measures will not be reached. And the current state assessment underestimates the risk hazardous substances posed to the marine environment. We include 15 substances or substance groups, and the other tools we have, for example, biological effects, are not really that developed so that it can cover all the other ones. So there is a need to develop biological effects as a tool for status assessment. And last but not least, we are always looking for experts to work within HELCOM and OSPAR. So if you would be interested in working with this, you can contact me and we can see if we can find something for you. That would be all. Thanks.
Okay, thank you so much, Johan, for an interesting presentation. Welcome, Joachim.
So I'm going to talk specifically about biological effect-based monitoring. We have an awful lot of monitoring in Sweden, a lot of programs for marine environment, for freshwater, for forest, for whatever. A lot of it is based on chemical analysis, as Johan said, where you can do chemical analysis compared to thresholds. Also in the European context, that's the common way of doing monitoring, I would say. A lot of the monitoring campaigns are very much based on chemical analysis. We want to work with biological effect more as some kind of integrated response to these cocktail of chemicals you have in the environment and look at health parameters. And we use a concept called biomarkers, just briefly going through that. And a biomarker, a biological effect marker, this is from my basic course, is a biological change, basically. You can have any biological system you want. A change in that you can link to a chemical exposure is a biomarker. So it can be like endocrine system, it can be growth, metabolism, detoxification systems. There's an awful lot of biological systems that can be affected, relatively specific by chemicals, and we use them as diagnostical tools. And a lot of work is to find and detect novel biomarkers to be used in assessments. And I mean, this work started in the 60s, 70s. We knew a lot about that chemicals had specific effects. And just in the history then, in the 80s, we had a Baltic Sea that was horrible. And the suspect was the pulp mill. And a lot of research groups start doing research to actually see if we could pinpoint pulp mill industry as the main source of pollution in the Baltic Sea. And I have an old colleague, Åke Bergman, that almost get a tear in his eye when he describes the Swedish EPA in the 80s as a hub for research, where different researchers from different disciplines could meet, exchange ideas and experiences, and then go out in the field and apply all these biological effect tools and chemical analysis tools and whatever they want to try. And they had like the Baltic Bay basically as an experimental lake. Well, sea, sorry. You can't say lake about the Baltic. They get really upset on the East Coast then. But anyway, so they did a lot of, this is just one example. This is a pulp mill Norrsundet, where they did a lot of work, and there's a very nice gradient from the pulp mill going up. So they could, and they sampled perch, but also a lot of samples for chemical analysis and so on. And they had a battery of, don't bother about the biomarkers, but there's a lot of different biological effects they would start looking at, and that they measured from growth to immune defense for detoxification enzymes, reproduction. And just quickly, don't get scared about this. Basically what they saw is in this gradient, the closer they came to the pulp mill, the more severe the effect were. The gonads went smaller, fewer white blood cells, detoxification enzymes went sky high, and so on and so on. So it was a kind of nice proof that you could use these biological effect markers as to identify a source of pollution, combined with chemical analysis. There was also a lot of that work done with population structures and ecology. From all this work, I think the Swedish EPA, well, if I heard correctly, kind of liked the approach, but they saw that all groups were kind of working scattered. Some were doing biological effect. As usual in academia, we all want to do our things in isolation. But so biological effects and chemistry there and population. So I think the Swedish EPA made an effort to gather these forces into an integrated program for coastal waters. That was at least the part where we work. And the integration was fish ecology, stock assessments, abundance, reproductive success. That was done by the fishery board in the old days, but SLU today. The individual health studies, biomarkers, that is basically Lars Ferlin that started that, and Åke Larsson here at GU drove a lot of this research. And then we have environmental chemistry that is administered by the Natural Historical Museum in Stockholm. So basically the strategy was to go out and sample at the same time and look at these different things, focusing on eel, pout and perch as two fish species. And we have had annual monitoring campaigns that started in '88, so we have really long time series. And the Swedish strategy has been since the beginning to go to this is a bit of a busy slide, but to go to unpolluted sites. Try to identify sites that are far away from anything, and the purpose then is to look at long time trend changes. Because if you go in and measure stuff in Gothenburg harbor, we know it's polluted. We don't need, I mean, it's obvious. So the idea was to try to identify long time effects or new effects in the environment by field observations. So we have these sites. Some, I think Kvädefjärd on the east coast and Fjällbacka are the two that from the beginning, from the '88. So we have really long time series there. So every year we've been measuring stuff there. Now, as Johan said, it's changing a bit the system, and this doesn't really meet the MSFD, because the MSFD want to look at good environmental status. So basically, we want to go into polluted sites to see how affected are they, and then with annual monitoring, see if it improves. That is the general idea. So what we are doing right now is adding also polluted sites to this to look to meet the MSFD better. But then also, just unfortunate cuts, we will need to change a bit of the sites coming here. But that has been the basis of our monitoring. And we have this battery of biological effect markers that we've been measuring forever then, that try to cover different functions in fish, like growth, reproduction, liver function, gene tox, oxygen transport, immune defense, and so on, to give like a overall picture of the health of the fish and to get the idea of the population health. That is the idea. And what we end up with is basically long time series. And you don't have to bother about exactly what it is. This is just one example. This is erode activity. Is an enzyme that metabolizes pHs, oils, and PCBs. And an increase of that is regarded as exposure to that kind of compounds. And we see slowly over the years an increase in the Baltic in several different sites, from Umeå down to Torhamn in Blekinge, in Kvädefjärden, all like in reference areas. So there is a slow change in the environment that we can detect with these markers. At the same time, we see a decrease in the size of the gonads, GSI, that's gonad somatic index in female fish. So the gonads are slowly decreasing over time as well. Also a long time series. And this is true for a lot of different biomarkers that we see. So we have a lot of results that is laying around. The one way to that it comes out is these faktablad that the SLU produces annually from different sites, but also combined for all sites or specific sites and so on. So there is a way to channelize to society the results, but a lot of the results stay a bit hidden, unfortunately. I'm coming back to that in the end. But we have been working a lot with this for many years now. We have a lot of experience when it comes to biological effect markers. But lately, last years, I've been more involved in a kind of European group, a European perspective. And in Europe, we have like some organizations that have working groups where I participate. We have the two conventions, OSPAR, Oslo Paris Convention, and HELCOM for the Baltic, and that, as Johan said, for Atlantic. And they have working groups deals with biological effects. The MIME, where I participate, is a working group on monitoring and/or trends and effects of substances in the marine environment. HELCOM have a biological effects subgroup that has been discussing this thing with biological effect markers for many, many years. Also, we have ICES, the International Council of the Exploration of the Sea. There is not like it's more like an organization that gives advice to these OSPAR and HELCOM and also governments or whoever wants. And ICES have a lot of working groups dealing mainly with fishery stocks, as you might know, but also for environmental chemistry. But there is one group, WGBAC, where I'm sitting as well, the working group on biological effects of contaminants, that has been dealing with biological effect markers for decades. And they've been doing a lot of assessment and going through the literature and proposing biomarkers forever to OSPAR and HELCOM, not being heard so much. Since I've been in these groups for some years now, in all these groups there's been a lot of discussions of the biological effect markers. How should they be used? And we should have an overview, but we're all doing it a bit differently. But basically almost the same bunch of people in different constellations doing almost the same kind of work in different ways. So it's kind of weird. So 2 years ago there was formed a group with the very horrible acronym SCEF, a study group organized by OSPAR and HELCOM joint. So it's two groups of 12 to 15 researchers from OSPAR, and they come basically from the WGBAC group in ICES, and another group from HELCOM. So the study group on developing new guidelines for the monitoring of biological effects of contaminants. And that was a very specific study group that had 2 years to deliver on a question that was asked by OSPAR and HELCOM joint. And the aim then of SCEF, there was a lot, but this is like a boiled down. It was to review and list or update a core set of relevant biological effect parameters that can be used already. And one important parameter is, as Johan said, was background criteria, BAC values, threshold values for biological effect. What is normal, basically, so we can detect a change. Update sampling strategies, and also sentinel species, and to optimize the current integrated biological effects approach in use and to propose new ones. So that was the main aims. So we've been working in different groups in this SCEF, HELCOM and OSPAR joint, to try to come with advice to HELCOM and OSPAR on how to use biological effect in a more structured and maybe mandatory way, for instance, the MSFD. It's been a very interesting experience, very democratic. So it's very easy when you start because you have the idea, so this can't be that difficult, but then you realize that people have different ideas than me. And that's sometimes disturbing. Or maybe good, I don't know. So just one outcome when it comes to biomarkers. This is a list we ended up after many discussions. Don't focus on the biomarkers themselves, but that we believe in that can be used, implemented now in monitoring. And that is based not on which one we believe are the best. The problem here is that we had to kill our darlings. I killed a lot of darlings, but some had more problems with that. But so there has been a lot of discussions. The criteria for this is that these markers have to have a clear link to a chemical exposure, and also that they need to have proper quality assurance made. I'm coming back to that, and that is ring test to demonstrate that different labs, when they measure it, get the same results. That's important. And we also need to have BAC values for the species where you use these background criteria. What is normal, like threshold values. So we came up with a suggestion, some for fish, bivalves, amphipods, and gastropods. And this JHS, these have been proposed before in different constellation in OSPAR and also WGBAC. So this work has been done before. But I think this time we really, but they'd be done in different like scattered groups mainly. Now this effort, SCEF, was really a joint endeavor with OSPAR and HELCOM combined. So I mean, we covered the field basically. So we have this, and basically what we will suggest is that this list will be, we will suggest that this will enter the OSPAR guidelines, and that member states should measure 3 or 5 out of these biomarkers that suits them. Because there is no way we can make them measure everything of this. But rather than picking out only 1 or 2 markers, we give them, these are good, and use 3 of those at least, and select those based on your experiences, ecology, or circumstances. And we also had a list of biomarkers to be considered, and then the other one that are like in a research state that we are working with. Another big discussion was the thing with sentinel species to work with. I had long discussions with a colleague if we could find one species that could be like one fish species for Europe, or at least for Portugal up to here. And there are a couple that you could maybe, but that was too difficult. And also here we have, I mean, in one way it would be nice if you could say, well, use DAB. Easy, everyone do that. But then most of the countries, I said Sweden, we have very long time series, and we have our history when it comes to monitoring. But most countries in Europe have similar development over but the programs are slightly different. The English do a lot of offshore trawling, looking at pathology offshore in DAB. The Germans do the same. The French, they do trawlings, but also these biomarker work. So all countries have their programs with long time series using this whatever species they started with. So of course you don't want to change. So we ended up, after many hours of discussion, coming to the conclusion that, yeah, use whatever species you want. But the important thing here then, if we will continue with this, we'll see what the response will be, is that we have intercalibration exercises. So we know if you have a response in one species, what does it mean in this other one? So we can compare at least. That I think that was what we ended in. So basically we will deliver to OSPAR. I said I'm sitting in a group called MIME, and you look at this convention, these organizations, the organization chart is very interesting. There are many boxes and arrows and stuff, and I had no idea where I was in this organization scheme. MIME group is very low, the working groups. Above us, we have a group called HASEC that can have, they can take like decisions in another way. So this HASEC group that is in OSPAR is the one that will receive our report now in March. It's March now, or no? Soon. No, yeah. So next month. So it's basically finished now, the report. And we will recommend these robust biomarkers that are ready to be implemented in monitoring based on, as I said, some criteria that we have back values, we have the link to chemistry and so on, and also that we have quality assurance, and then we have candidate biomarkers, and we will also list the strategies for Q&Ing candidate biomarkers, how to do ring tests, and sentinel species and monitoring strategies. And this is, and I think the main aim is. OSPAR have this SEMP list, it's called Coordinated Environmental Monitoring Program. And I have a colleague that I started in the MIME group because more organized because a colleague of ours retired, Thierry, and then his colleague Aurel asked me to take his place. And she's been talking since the beginning that we need to push biomarkers into the SEMP list. And I never really understood what she meant. But basically that's what it is, to get HASEC to say, yeah, these are robust, we believe in this, so this will be a part of this SEMP, the Coordinated Environmental Monitoring Program, making it a bit more mandatory. I hope I'm correct now, Johan? Yes. For the member states to actually include that in the monitoring programs. At the moment there is one marker actually in SEMP list, and that is imposex in snails, because that's a very easy, clear biomarker that is so clearly linked to TBT, So it was quite easy to get that one in. So our aim has been then to push, as we say, this list of biomarkers into the SEMP list. 3 biomarkers, 3 minutes. Oh, well, this is going better than I thought. How nice. So that's the main aim. So, but it's been, I mean, behind this work, we have had at least 30 years of discussions. So things go very slowly. So now, at least now the MIME group supported this, and they were happy with our report and the strategy. So we got full support from MIME and also from HELCOM. So now the next level is then HASEC, the ones above, whoever that is, that will decide if this is. But we are going away, and I think the use of biological effect markers is not the only thing that we should do, of course. But I think it's a nice complement to the chemical analysis we do today, because I think with just analyzing chemicals, we miss a lot of things. And I think monitoring is important. And something we sometimes forget is that we talk about monitoring. We are so focused on seeing if something is bad in the environment. But biological effect monitoring is also a very nice way to show that things improve. As we did see in the Baltic, when they start to be working with that processed water and all, and in Stenungsund, we've seen improvements since the 90s when I started looking there. So it's a nice way to see in fish, in the fish health, that things are getting better if you take measures and improve whatever you're letting out. So what we are now, we deliver this report, but we are also continuing the work in different constellations, doing proficiency tests, ring tests for markers. So I spent half of January sending boxes of dry ice with samples out to different labs in Europe. It's a horrible experience. We're upgrading, standardizing protocols. ICEs have something called a time series from a lot of these biomarkers. So based on the ring test, we will see how they perform, and then we will update a lot of these protocols. And we will also have exercises where we establish background assessment criteria, back values, for species that doesn't have it yet, that is necessary for assessments. And that will be done hopefully also with some practical workshops where we get, because a lot of people doing these ring tests, there are a lot of labs that want to start. But I mean, it's very easy to measure, let's say, an enzyme called acetylcholine esterase. If you've done it a lot, it's not a problem, it's very easy. My students does it. But if you are in a lab that never done it before, there is a lot of things that can go wrong. So these practical workshops are very good to, if you want to kind of spread know-how in an efficient way. And then finally, and just to shock you a bit with color in this monochrome presentation, I nicked this from a colleague's PowerPoint. This has been also discussion in SCEF, but it's also been a parallel discussion for some years. And that is touching on the thing I said before with a lot of data. We have a lot of data in our monitoring program. We have biomarker, we have chemistry, and they're sitting in databases. And we're kind of looking at it individually. But there is a need of methods to integrate data, biological effect data with chemical data. Basically, to take a lot of complex data and boil it down to green, yellow, red. If you have a site, you integrate the data. And this is just one. There are many different ideas on how to do this now, flying around. This is from a colleague in Italy, Francesco Regoli. He's doing a lot of statistics, and he had this weight of evidence approach, where he wanted to have chemical analysis, how bioavailable it is. There are biomarkers. He wants to squeeze in bioassays as well. We'll see how that goes. But also combine that with meiofauna and biodiversity, basically, and try to find models, statistical models, to integrate all this data into some kind of scale that says something about the state of that specific site. And that would be one nice way to use all the data in such a way that is easy to present to authorities and so on. So we just got money from Naturvårdsverket, actually, a project for monitoring, where I can hire a postdoc to do that. Well, maybe not that, but something similar. So that was it for for today. Thank you very much. This is a slide I stole from my old supervisor, Lars Ferlin. Because it's very nice to do monitoring, because you get to go to these sites. Thank you very much.